Now this particular operon is also controlled positively, so it has both negative and positive regulation. And positive regulation is carried on by a protein that's called the cap protein. And the cap protein stands for lots of things. It's called cyclic AMP dependent activating protein. That's why it's CAP. It's called a catabolic activating protein. Sometimes it's called CRP, catabolic repressor protein, different things like that. But the cap protein, right, has a region that it binds. So here's our gene that we talked about. Here's the promoter region here. And notice this thing here, polymerase binds. Remember we said polymerase has a pretty big footprint. Even in E. coli, the footprint, right, if this is the binding site for polymerase here, then it has, a footprint of, what is that, 80 nucleotides or so, 80 base pairs. The operator is right in the middle of that. So when it binds, when the protein, the repressor binds, it blocks that footprint. The cap binding site, or the cap protein, binds at a site. It's a specific sequence, once again. These proteins bind and recognize specific site sequences. So the cap protein binds to that sequence. And what the cap protein does is it activates transcription. Because you notice the cap protein does not overlap with its polymerase binding. Right, here's RNA polymerase. You can see it bound all in this site here, covering everything, including the region where the repressor binds. But the cap protein doesn't overlap. In fact, it helps to recruit polymerase because it has a region of the protein that actually interacts with the polymerase so that it draws or pulls the polymerase into the site actively. So it's an activator, a repressor and an activator. The repressor protein negatively regulates expression. That cap protein actively regulates expression. Between the two of those, they determine the behavior of this particular gene or the transcription of this gene under situations where there's either glucose and lactose, both of them, right? Here's an example. If there's both glucose and lactose there, if there's lactose there, it binds to the repressor protein, so the repressor protein can't bind to the repressor. If there's glucose present, if there's a lot of glucose in the cell, then you don't have to activate this thing, right? You don't have to activate this pathway. So the operon is off because the cap protein is not bound. In the absence of glucose, the cap protein doesn't bind. Remember, the cap is an activator of the lactose gene. I don't know if I have a slide that tells you I don't have one. No. So, let me see if I can tell. Okay, so this cap protein is an activator, right, for the lactose operon. Cap protein, remember, is a cyclic AMP dependent protein, so it has to bind cyclic AMP. And when these cells when there's glucose present in the cell, right, the cell makes a lot of cyclic AMP. So one of the products of having glucose in the cell is the production of cyclic AMP. That makes sense? So it produces cyclic AMP, it's the other way around. When there's glucose in the cell, cyclic AMP levels go down. So there's no cyclic AMP to bind to the cap protein to activate the cap protein, so the cap protein doesn't bind to the promoter of the lac operon. Does everybody follow that? I'll say it again. Okay? So why does that? Remember, you got to think about what's happening, right? If the cell is in glucose and lactose, it prefers to grow on glucose. So you want to turn this lac operon off. In the presence of glucose, there's no cyclic AMP. It doesn't bind to the cap protein. The cap protein is not active, so the cap protein doesn't bind to that site. So in this condition, plenty of glucose, plenty of lactose, right? There's no the cap protein doesn't bind to this promoter, so it doesn't activate it positively. There's lactose there, so nothing's here. But anyway, this guy's turned off because there's no cap protein now. Okay? Now here's in the cells in glucose and no lactose. Well, in the absence of lactose, there's no lactose bound to the repressor. The repressor binds. There's no cyclic AMP, so the cap protein doesn't bind. So this is also turned off. It's turned off, double turned off. There's no cap protein, so we're not initiation. and their repressor protein sitting there. So now we come to the situation, no glucose, no lactose. No lactose, the repressor sits there. No glucose, the cap protein sits on. But the operon is still turned off because the cap protein says there's no glucose there, right? We need to turn this operon on. But this repressor protein says don't bother, there's no lactose either. So in the absence of glucose or lactose, the thing is shut off. But now in a situation where there's no glucose and there is lactose, there's no glucose, cyclic AMP levels are high, right? They bind to the cap protein, which activates it. The cyclic AMP actually causes it to form the tetramer. It then binds to the cap binding site, and it positively influences transcription. There is lactose. The lactose binds to the repressor, so the repressor falls off. There's no repressor on the repressor site, so the combination of the two of those turns it on. So by having positive and negative regulation, this operon can determine not only whether there's, if there's lactose there, but whether there's glucose there, and it shouldn't use the lactose, right? In all situations, or whether there isn't any lactose, there isn't any glucose there, right? Or lactose there, and it just doesn't work either. Make sense? Question? If you just look at it and think about it, it's actually pretty easy. So this is just to show you the cat binding protein, right? It's called the catavalproic activating protein. Sometimes it's called the CRP, which is the cyclic AMP receptor protein, because the cap protein binds cyclic AMP, so it's called the cyclic AMP receptor protein. When it binds cyclic AMP, then it becomes activated. And this just shows that there's your DNA strand again, and it shows how this protein binds. And the reason I put this there more than anything is to show you that all of these red amino acids are charged amino acids. And these charged amino acids are interacting, or they're forming hydrogen bonds with the bases that are wrapped inside this thing. So the sequence in here is the cap binding sequence, and the cap binding protein recognizes and binds to it very tightly. And what it does is helping to recruit proteins over to this side here. Okay. We've talked about this pretty much. This is just another slide that shows you negative control and positive control and how it works, right? Here's the repressor, and it binds there. And if you add the inducer, it pulls it off. So this is negatively controlling. On the other hand, this is positive control, where there's an inactive inducer, and it binds the inducer protein, and that causes it to bind and activate. So it activates here. So this is like the cap protein. Cyclic AMP binds to the cap protein, causes it to form tetramer, which is then able to bind to that cap site, which will allow polymerase to bind and go. On the other hand, this is the repressor. So it shows the same thing we talked about, just described it a little better. We've been talking about chromatin. Here's a picture of what we talked about. I want to show you a little more about these nucleosomes and how they work, and one of their functions, which is to wrap that DNA very tightly. This is the so-called string of beads. You can see that this string of beads, well, you can't see, they're about 100 angstroms. The strand in between is the linker DNA, which is about 10 angstroms. And that DNA comes, wraps around this guy twice, and there's a bridge between it of 50 nucleotides, and then wraps again, and wraps again. You can see how uniform it is. This DNA then will wrap up into a coil, a supercoil, or a spring almost, right? And if you take this and just hold it steady and kind of wrap it, twist it, it'll form this. So you can see there's individual nucleosomes that natively stained, so the white is actually the nucleosomes. This thing is about 300 angstroms across. One of the functions of this, as we said before, is to compact DNA. In mammalian cells, there's 2,000 times more DNA than in E. coli, so it has to be compacted. And this, when you do this, it compacts it about 7-fold, and when you do this, about 7-fold more again. So you shrink it more and more and more. I told you we talked about that. I'm not going to show you again. I want to tell you a little more about the histone modification. Not a whole bunch. We talked a little bit about acetylation. But if you look at the histones, you can see there's that tail again, the N-terminal tail, the amino-terminal domain. There are lysine that can be phosphorylated. Right? So these guys can be phosphorylated. And then there are some of these groups also can be methylated. Here it tells you once again about the HAPs and the histone deacetylases, so it's written down as you can see that. Acetylation removes positive charges, and deacetylation does exactly the opposite. Now, you hear about it, we're not going to talk about, you will in genetics, you will in cell biology, you can talk about inheritance, right, which is not related to the DNA itself. So cells can inherit characteristics which are not directly represented in nucleotide in base sequences. And they're able to do that because some of these modifications can be passed on from cell generation to cell generation. In particular, you've heard about methylation. You can develop a table from this thing, and I don't know if I put one in there or not. Yeah, here's one. If not, there's one in your book. But we know people are beginning to map what happens to these things and to these different modification sites. They're not all modified at the same time. For instance, the acetylation of particular the lysine number 9 in the H3 histone, right, is associated with the formation of nucleosomes, the deposition. So during DNA synthesis, when new nucleosomes are being added, this guy's acetylated. If you look there, you look at gene expression, right? If you look at modifications, the acetylation of histone of lysine 14 on the H3 histone, and the phosphorylation of the serine, right, at position 9, lead to expression. So there are different. We are starting to map these things into modifications, and to look at specific acetylations and methylations, which either activate transcription or silence transcription. If you look at some of these that say silencing, gene silencing, right? Unmodified histone H4 is silencing. But also, in general, this methylation, in particular when it's alone, so in histones, if you methylate 9 rather, or I guess 9 rather than acetylate it, it's associated with silencing of chromatin, silencing of expression. And you realize that silencing of expression has to do with taking chromatin and making it even more tightly bound, right? So we talked about the tightening of DNA around chromatin and how that is regulated or regulates expression. If you make it even tighter, it regulates even more. So over the years, actually it's been for 50 years, people have identified or have recognized that genes that are not being expressed in cells are very highly methylated. Right? And methylation is associated with non-expression. Very highly methylated DNA forms what we call heterochromatin. And heterochromatin is very, very tightly packed chromatin, which enzymes have no access to, and that chromatin is not transcribed. Okay? Very tightly packed. And this methylation of these tails is associated with the formation of chromatin. And that can be inherited, right? Because when DNA replicates, there are enzymes which recognize these methylated regions, methylated nucleotides, and they'll methylate them again in the new cells so that it shuts off expression. So methylation is very, very important. Okay, that's all I'll say on that. Okay. So we talked a little bit about